Hello everyone, good morning. Today we are going to discuss about mechanism of fixation. So in this video we are going to discuss how the fixative fix the disease. So the wet fixative usually work as the dehydration and coagulation of protein and cross-linking the fixative. So in here, when we consider the fixatives, two wet fixatives, they act as two methods. One is dehydrating, remove the water and coagulate the protein. Another method, cross-linking the fixative. So now we will look at the dehydration and coagulation of protein. And the methanol and ethanols are commonly used coagulative fixatives. So these two alcohol remove the water from the tissue and causing destabilization of hydrogen bond and thereby disruption of the tertiary structure of the protein. However, the secondary structure of the protein is maintained. Ethanol is relatively stronger dehydrating agent than the methanol. That's why we are using the process of the tissue processing. We are using ethanol. The ethanol and the methanol start work from 60% to 80% concentration. So normally we have to prepare the series normally 60%, 70, 80 we are using for methanol and ethanol concentration. So disadvantage, when we consider the disadvantage of the dehydrating fixatives, there are two. One is shrinkage of the cell. Other one is the removal of the soluble substance from the tissue. These are the disadvantages of the dehydrating fixative. Other one, cross-linking fixative. So here we are using formaldehyde, glutaraldehyde, osmium tetroxide for the cross-linking fixative. So when we consider the formaldehyde, formaldehyde in aqueous solution combined with the water to form a methylene hydrate, a methylene glycol. So the first picture here you can see the formaldehyde treated with the water and it produces the methylene glycol. Then in a long-standing position, this methylene glycol may further react with the water molecule and form a polymer known as a polyoxymethylene glycol. So, these again depolymerized in the methylene glycol in a neutral buffer system. So in here what happened, after producing the formaldehyde treated with the water and it's produced methylene glycol. This methylene glycol again and again react with the water molecule, so it produced polyoxy methylene glycols. Then it is this polyoxy methylene glycol polymerize and it produce the methylene glycol, right, when the presence of neutral buffer. The formaldehyde react with the various side chain of the protein and the form hydroxymethyl side groups. So it is the again formaldehyde react with the side chain of the protein and it's produce the hydroxymethyl group. These compounds are highly reactive and subsequently cross-linking occur by forming the methylene bridge. This preliminary reaction of hydroxymethyl side chain is the primary reaction, and the subsequent intermolecular and intramolecular cross-linking of the molecules occur as a slow growing process. This ultimately produce an insoluble product. So the formalin can be removed from tissue by prolonged washing. However, once methylene bridge is formed in the tissue, the reaction is stable and it is difficult to remove formalin from the tissue. Formaldehyde also reacts with the nucleic acid by reacting with the amino group of the nucleotide. Then we will move into the glutaraldehyde. It has two aldehyde groups that are separated by three methylene bridges. The aldehyde group of the glutaraldehyde react with the amino group of the protein, predominantly lysine. When one aldehyde group reacts with the amino group, the other free aldehyde group may help to cross-link. The glutaraldehyde rapidly and irreversibly cross-link the protein. The penetration of glutaraldehyde is lower than the formaldehyde. Osmium tetroxide. Osmium tetroxide is mainly used as a fixative in electron microscope. So it is used alone or as a combination with other agents. So osmium tetroxide react with two unsaturated carbon atom of the lipid and cross-link with them. The compound causes oxidation of unsaturated bond in the biological tissue, particularly lipid. It converts the unsaturated fatty acid into a stable product known as a glycol osmate. The tetravalent osmium becoming a hexavalent in this reaction. Osmic acid monoester forms in this reaction is easily hydrolyzed to a diol and osmic acid. Osmium tetroxide may react with the two unsaturated carbon atom of lipids and may cross-link. So this is the summary. Here you can see the picture. Normally the osmium tetroxide, it is tetravalent 4, then it is react with the lipid molecule, unsaturated lipid molecule. Unsaturated means it's contain double bonds. So this double is the osmium tetroxide react with a unsaturated fatty acid, that means double bonds. It's form hexavalent osmium after reaction. You can see here the hexavalent 6. Then now the osmium oxidation number is 6. So here the osmium tetroxide is oxidizing and the unsaturated fatty acids are reduced number. Then these hexavalent osmium react with water molecule and it's produced diol and osmium trioxide. So this is osmium trioxide and diols. These are react with molecule and its form or it's cross-link the the fatty acids and cross-linking each other. So the mechanism of different fixative here. First one, alcohol works by removing water from the tissues and coagulating the protein. This is the alcohol, how they fix the tissues. Second one, formaldehyde. It's form the hydroxymethyl side groups and cross-link with the protein. Osmium tetroxide forms glycol osmate with the lipid molecule. So first alcohol and formaldehyde, they react with the protein, but osmium tetroxide, they are react with the lipid molecule. So these are the references.